Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs for the year 2020. I will be discussing the applied biology and methodology section. If you are new to my channel, please do like, share and subscribe. Then next one. In a cloning experiment, a DNA molecule isolated as a PST1 fragment was inserted into the PST1 cloning site of a cloning vector that is 3 kb in length. Digestion of the confirmed clones with PST1 resulted in the appearance of a single band corresponding to 3 kb in an agarose gel. Based on this information, what is the probable size of the cloned fragment? So usually in restriction digestion, you will get a band of 3 kb insert and a band of the insert as well as of the vector. But here the vector and the insert both are 3 kb full length. So you will get only a single band of 3 kb. So the answer here is option 3. Then next one. Which of the following statement is true? Nick translation cannot be used for radio labeling DNA fragments for use in southern hybridization. That is not correct. RACE can be used to obtain sequence information of the ends of cDNA. That is correct. In a 2D gel electrophoresis for separating protein, analytes are first separated on the basis of their size and then on the basis of their pI. It is first on the basis of their pI and then on the basis of their size. So 3 is also not correct. AFLP markers are typically used to screen heterozygotes from homozygotes. AFLP is a dominant marker, so it cannot be used to screen heterozygotes. So the answer here is option 2. Then next one. The following disulfide bond containing peptide was digested using trypsin. How many peptide fragments will be produced by the digestion? So trypsin digests at the carboxyl terminal of lysine or C-terminal side of lysine and arginine. If lysine or arginine is followed by a proline, then it will not cut, and trypsin, it is an endopeptidase, so it will not cut at the ends. Now if you search for lysine and arginine in the sequence, you will find one over here, and that is lysine. And here also one lysine is there, and here also one arginine is there. So it will cut over here, but since there are disulfide linkages in this one, it will be separated as a single peptide, and this will be your third peptide. So so how many fragments you will get? You will get 3 fragments. So the answer here is option 1. Next question. A protein found in the Golgi compartment was found to be degraded rapidly at mitosis. The Golgi extracts isolated from G1 and M phase cells treated with de-ubiquitination inhibitors. So those which remove ubiquitin molecule have been blocked. So ubiquitin will be attached once you use de-ubiquitination inhibitors. Were treated with glycosidase. Now why treatment with glycosidase? Because Golgi apparatus, there is O-linked glycosylation taking place. So protein molecules have glycans attached to it. And Western blots done using antibodies against the protein. The following pattern was obtained. Identify the statements that cannot be made based on the data. So if you see the G1 phase, there is minus glycosidase. There is no glycosidase is added. So glycan molecule is attached and it moves, this band moves slowly than the one in second lane, in which glycosidase has been given and glycan molecules have been removed from the protein. So it is running faster. But in case of minus glycosidase, this is running slower because the glycan molecules have not been removed. Similarly, here also in case of M phase, where glycosidase has been treated, the band runs faster than the one where glycosidase have not been treated. Then these too many bands over here, they represent ubiquitination because ubiquitin molecules attach non-uniformly. And in G1, ubiquitination doesn't take place, whereas in M phase, ubiquitination takes place. So coming to the statements, they have asked the statement that cannot be made based on the data. The protein is glycosylated in G1 and M phase. That is correct. The protein is glycosylated and ubiquitinated in M phase. That is also correct. The protein is ubiquitinated but not glycosylated in M phase. That is not correct. It is glycosylated in M phase. The protein is not ubiquitinated in G1 phase. That is also correct. So the correct answer here is option 3. Then next one. A group of researchers are testing two new agents, M1 and M2, for their efficacy in selecting transgenic plants. When they performed tissue culture experiments using 3 explants A, B, C without agrobacterium transformation and selected the regenerated plants on M1 and M2, the following regeneration frequencies were obtained. So this such kind of experiment where without treatment with agrobacterium transformation, you select the plants on antibiotic medium of different concentration. So whichever concentration there is death, it will be a death of the explant. That concentration will be used for selection after agrobacterium transformation, because after agrobacterium transformation the antibiotic resistance gene is already present, and at that concentration where the death is taking place, the plant will be able to regenerate. So we will see, based on the above data, which one of the following conclusion is incorrect. M2 is a stronger selection agent than M1 for explant types A and B. So that is correct, because you are getting more death in case of M2 than in M1. So option 1 is correct. A concentration of 100 mg per liter of M1 and 15 mg per liter of M2 can be used for selection of transformed cells using explant type A. So this is A at 100 mg, yes, in case of M1, and at 15 mg in case of M2. So that statement is also correct. Among the 3 explants, type C is least suitable at the tested concentrations of M1 and M2. That is also correct, because there are no death in case of C. Then next one. Use of M1 with explant type B is the most ideal combination for selection of transformants. Use of M1 with explant type B. No, use of M2 with explant type B. That is correct. So 4th is not correct. So 4th is the incorrect conclusion. So answer here is option 4. Then next one. Given below is a schematic representation of few restriction sites in the multiple cloning site of a plasmid. HIN3 XHO SMA1 PST1. The recognition sequences of the above enzyme and their digestion pattern are shown below. Which of the following combination of enzymes from the vectors can be used for cloning a DNA fragment obtained as SAL1 HIND3? So SAL1 is a staggered end cutter or sticky end cutter, and HIN3 is a blunt end cutter. So SAL1, I already told you it is XHO1. That I have already discussed once in one of my videos, where the question regarding SAL1 and XHO1 was asked. So you can go through the discussion over there. So XHO1 and SMA1. SMA1 is a blunt end cutter. So the answer here is option 3, that is XHO and SMA1. The question was almost similar to this question. You can refer to that video. Next one. Given below is a schematic representation of the tDNA region of a transgene construct and the southern blot analysis using PST1 digested genomic DNA of 5 independent transgenic lines labeled A to E developed using the construct. So this is the probe region, and this is the southern blot diagram. The probe used for hybridization is shown as black box below the construct. Based on the above data, which one of the following option gives the correct list of single insertion transgenic events? Now if you see this diagram, this is and these, the bands are not variable, but the between ones, they are variable. So we will see the ones which are in between these two bands. So this one, A is having a single copy insertion. B is also having only single band, so single copy insertion. C is having two bands, so that is double copy insertion. D is having three bands, so it is triple copy insertion. And E is having a single band, so that is also single copy insertion. So the answer here is they have asked for single insertion transgenic events. So it is A, B, E only. The next one. Given below are the restriction profiles obtained upon digestion of a 4.8 kb plasmid with 4 different enzymes E1, E2, E3, E4. So E1 4.8 kb, E1 plus E2 500 base pair plus 4.3 kb. Then E2 4.8 kb, E2 plus E3 400 base pair plus 4.4 kb, E3 4.8 kb, E3 plus E4 2 kb plus 2.8 kb, E4 4.8 kb, E1 plus E4 1.9 kb plus 2.9 kb. Based on the above information, which one of the following statement is correct? E1 and E4 have two recognition sites each in the plasmid. I have drawn the diagram in the next slide. 1 minute. Okay. So this is E1, E2, and they have told that E1 and E4 have got two recognition sites each in the plasmid. Okay, before that, E1 plus E2, E1 and E2, if you have E1 over here, E2 over here, you will get a 0.5 kb band over here and a 4.3 kb band over here. So totally 4.8 kb. Now in the second case, E2 plus E3, E2 plus E3, you will get E2 will be in the center of E1 and E3. E2 will be flanked by E1 and E3. Only then you will get a 0.4 kb or 400 base pair band and a 4.4 kb vector band. Now E3 and E4, you know where the position of E3 and E4. So here you will get a 2 kb band and a 2.8 kb band. Now E1 and E4, E1 and E4, this will be 1.9 kb and this will be 2.9 kb band. So now we will see the information or statements they have given. So E1 and E4 have two recognition sites each in the plasmid. E1 and E4 have got only single recognition sites in the plasmid, not two. Then E2 recognition sequence is located between E1 and E3. That is correct as per this figure. The sequence of recognition sites after digestion of the plasmid with E3 is E4, E2, E1. After digestion with E3, you will get E2, E1, E4. That is not correct. Then the sequence of recognition site after digestion of the plasmid with E2 is E1, E3 and E4. That is also not correct. So the answer here is option 2. E2 recognition sequence is located between E1 and E3. So coming to the year November 2020, shift 2. A panel of 6 hybrid cell lines, each containing a different subset of human chromosome, was examined for the presence of the gene product as shown below. The gene which codes for the given gene product is located on which chromosome? So this pattern has to match with one of the chromosomes over here. So if you see, it is A plus B plus. A plus B plus you can see in chromosome 4 plus A plus and B plus. C D E minus. C D E minus in chromosome 4. F is plus. Chromosome 4 also F is plus. So the gene which codes for the given gene product is located on chromosome 4. Answer is option 4. Next one. In agrobacterium mediated transformation, which one of the following approaches is more likely to generate transgenic plant with incomplete transfer of the passenger gene? So placement of selection marker gene towards left border, no. And passenger gene towards right border, no. Placement of selection marker gene towards the right border and passenger gene towards the left border will give you incomplete transfer. So the answer here is option 3, that is placement of passenger gene towards left border and marker gene towards right border of tDNA. Then next one. Given below is a schematic representation of a southern blot performed to identify single copy integration events of the tDNA among 6 transgenic plants T1 to T6. Which one of the following options represent potential single copy events? So if you see this given blot, these are the untransformed controls. Now they are not variable. Now if you see T1, it is having only single copy transgene, only one single band. T2 is having two bands. So it is a double copy integration. And this one, T3 is having 2 copies. Then T4 is having 1, 2 and 3 copies. T5 again 1, T6 again 1. So T1, T5 and T6. They are having single copy events. So the answer here is option 1. Then next one. Expression of a gene A is regulated by Mg2 positive. They have just told it is regulated by Mg2 positive. The expression of gene A in the untreated and cells treated with Mg2 positive was analyzed by northern hybridization, or northern hybridization is also called northern blotting and western blotting. A similar exercise was done for the mutant, which was isolated with a 6 base pair deletion in 5' UTR of the transcript of a gene A. The following are summary of 4 possible results that are hypothesized to be obtained. Now expression of A, it is regulated by Mg2 positive. Okay. There are 2 types of cells, that is untreated cells and treated cells with Mg2 positive. Then they both were analyzed by northern hybridization, which tells about the RNA level, and western blotting, which tell about the protein levels. Then they have done a work on mutant also, which was isolated with 6 base pair deletion in the 5' UTR of a transcript of the gene A. So the following are summary of 4 possible results that are hypothesized to be obtained. Now they have told that if the regulation of gene A expression is controlled only at the level of translation, that means your RNA is not going to change in treated or untreated cells, because Mg2 positive act only at the level of translation. Which of the above profiles are possible correct representation of the experimental result? So if you see option B, in northern blotting, there are bands for RNA, but there are no bands for northern blotting in case of mutant. So that is not correct, because It is already told in the question that gene A expression is controlled only at the level of translation. So your mRNA is going to be produced whatever be the cause, whether you add Mg2 positive or whether you don't add Mg2 positive, mRNA will be produced and you will get bands in your northern blot. C also similarly, there are no bands for untreated sample of in northern blotting. So that is also not correct. C is also not correct. But if you see A and D, A is having band for RNA northern blotting in untreated and treated sample, both in mutant and wild type. Similarly, here also northern blotting is having bands in treated and untreated samples. Now coming to the western blotting. Western blotting, they have told that Mg2 is regulating at the translational level, that is at the protein level. So if you see the first case here, Mg2 positive is added, then translation take place. When Mg2 positive was added, translation take place over here. That is why you got a band for treated samples but not for untreated samples. Then if you see option D, for untreated sample you got a band but you didn't get a band for treated samples. So here they have only mentioned in the question that expression A is regulated by Mg2 positive. How Mg2 positive regulate translation, or whether they have not told whether Mg2 positive if present will favor translation and if absent will not favor translation, or Mg2 positive if present will not favor translation, or Mg2 positive and absent will favor translation. They have not told anything like that regarding Mg2 positive, how it is regulating translation. So both A and D will be your correct answer. A and D. So the answer here is option 3, A and D. Now next one. An experiment was performed to introduce a transgenic trait in a crop plant by agrobacterium mediated transformation using a transgene construct in which the transgene was expressed using the CAMV35S promoter. It was observed that expression level of the transgenic protein were very low in all transgenic plants, while transgene mRNA levels were high and variable among different plants. Further, different transgenic lines contain different numbers of the tDNA insert. The following statements were made to explain the above observation. Now, what they have done was they have introduced a transgenic trait, but they have not mentioned the source from where the transgenic trait was taken. And it was introduced into crop plant by agrobacterium mediated transformation. The transgene construct was placed under the CAMV35S promoter. So it is a constitutive promoter and a strong promoter. Then and it was observed that expression level of the transgenic proteins were very low. The protein level was very low, but in the transgenic plants, while the transgene mRNA levels were high and variable among different plants. Further, different transgenic lines contained different number of the tDNA insert. The following statements were made to explain the above observation. Now, I have already explained a similar question in one of the videos where they have asked regarding bacterial and human codons. That was based on codon bias. So here also the answer is the same thing, that is why you get different low level of protein expression, maybe because the transgenic trait was taken from bacteria and the bacterial codon is different for when compared to the codon which that plant uses because of codon bias. You can go through the video previously where I have done regarding codon bias. So answer here is low expression level of the transgenic protein in all transgenic plants could be due to codon usage variation between the host plant and the heterologous source of the transgene. That is what they have told. There is codon usage variation between, because for if it is suppose the codon for arginine is GUA, that is being used by the bacterial gene for expression in the bacterial expression in bacteria, but GUA is not the codon for arginine in case of the host plant, so it cannot recognize the GUA codon as arginine codon, and the protein expression will either be hindered over there or it will be stopped there. So you will get very less levels of that protein in the transgenic plant. So the answer here is option B. So answer is B only. Then next one. Given below are statements related to various molecular techniques. Which one of the following option represents a combination of correct statements? During molecular cloning of DNA fragments, a vector and insert molecule digested with two different enzymes can never be ligated with each other. They can be ligated, so that is not correct. A vector and insert molecule digested with two different enzymes can never be ligated with each other. They can be ligated. Only 3' 5' exonuclease and not 5' 3' exonuclease can be used for digesting nucleic acid to generate blunt end fragments for cloning. That is also not correct. In Sanger's dideoxy sequencing method, each reaction consists of a mixture of 3 DNTPs and 1 DDNTP. That is correct. Self-ligation of a vector with compatible ends can be prevented by treatment with alkaline phosphatase. That is also correct. So C and D are correct. The answer is option 3. Then next one. Given below are 4 DNA sequences and set of forward and reverse primers for PCR amplification. In the absence of any other factors but not restricted to TM length and percent GC etc., which one of the above template primer combination would produce an amplified fragment? So here the forward primer will be same as this sequence, and the reverse primer will be complementary to this, but you have to read it in the 5' to 3' direction. Now if you see in the case of option A, the forward primer is not the same as this sequence, so it is not correct. Similarly, B also, the forward primer is not the same as this sequence, so it is not correct. Now if you take C, if you see, it is CTTGA CTA. So CTTGAC, that is correct. Now reverse primer is complement of A is T, C, G, TA, GC, AT, CG. So if you read it in the 5' 3' direction, it is TGACTG. So that is correct. C is correct. D, if you see, okay, the forward primer is correct, but the reverse primer is CAG. It is GTC. So that is not correct. So the answer that is correct here is only option C. That is C only.